All right, hi everybody, thanks for coming. I'm really pleased to introduce Dr. Joel Swift, who's been a postdoc associated with my lab for a few years. So Joel began his scientific career at State Fair Community College.
Roadrunners.
Go Roadrunners. Followed by University of Central Missouri, where he got his degree in integrative biology, the best kind of biology. And then he went on to work as a technician at the Missouri Botanical Garden for a while in conservation genetics. After that, he did his PhD at St. Louis University in the lab of Allison Miller, who many of us know and get to collaborate with, studying the microbiomes of grapevines. And after that, he joined us at KU at first, working on a project to identify groups of bacteria that would improve drought tolerance in corn. That was funded by the USDA. And then in 2023, he began working on comparative studies of corn and Tripsacum dactyloides, which he's going to be talking about now, with the goal of understanding how these plants resist flood stress and drought stress. And that work is supported by the National Science Foundation. Joel is a NSF Postdoctoral Fellow there. So Joel, take it away. I'm excited to see what you have to tell us.
Thank you. So I'm from Missouri originally, and now I'm working in Kansas, but I've always been at awe in prairie ecosystems for the resilience, and I think they're just stunningly beautiful, especially this shot from one of our former postdocs. Looks like it's in the early spring or maybe in the beginning of the summer. We can see all these different plant species. But when I'm driving around where I'm from, this is what I see. I see maize everywhere, or soybeans, depending upon the year. So I've always thought, we have these very resilient prairie ecosystems that we've mostly, unfortunately, destroyed. But what can they tell us about how we can, improve yield, improve productivity of maize so we can grow less of it, or we can grow more of it on less acreage, is what I'm always thinking, because a lot of these lands have been degraded by farming, but I'm hopeful that if we can get more out of the acreage we do use, maybe we can further improve. So maize productivity, it's no secret that it's vulnerable to the stresses that we see on a yearly basis or over a series of years. So I've got a picture of drought stress affecting maize out in Kansas, as well as this picture here that's showing the detrimental effects of flooding on maize. Particularly, this is maize at kind of a later point, but it's really vulnerable to waterlogging at the early points of the season when you're planting in the spring. So if you get a ton of water early in the maize's life cycle, you're going to get a ton of crazy kind of phenotypes that are going to lead to lower yields. A lot of issues with nitrogen uptake as well after flooding. But when we look to prairie ecosystems, there's a lot of prairie plant species that are able to survive these types of environmental stressors. So there's a salvia over there, and you can kind of tell that prairie looks very parched, very brown and dry. So it's experiencing kind of this drought, and we can see, pop-ups of green of this kind of salvia that's growing, rather unabated by this particular drought, as well as flooding is kind of common in some prairie ecosystems where we see either seasonal or maybe temporary inundation of areas of the prairie. So there's species that are more adapted to kind of a riparian environment in these particular prairies. And so the reason why we look at Tripsacum dactyloides is that when we think about maize and where we're growing maize here in the temperate regions, this little thing, I guess if somebody wants this citation, let me know, but I'll try to keep this box out of the way. When we think about where maize was domesticated in zea diversity, it's concentrated in the tropics, yet we grow maize in the temperate regions of North America and across the globe as a whole in many temperate regions. So if we look and kind of expand our view of the diversity of maize and some of its sister genera, we can see that we have accessions or species that grow across much wider areas of the planet. So we've got Tripsacum dactyloides here, which grows out of the in the tropical regions and then up into the temperate regions of North America too. So we're thinking that as far as comparing this, it's maize's most temperate relative. It would make a good proxy to understand how we could adapt maize to these more temperate environments. And also, many individuals have looked at Tripsacum is a source of novel genetic diversity to intergress into maize. So this is Tripsacum here. This is a very picturesque and very nice herbarium specimen. Anybody who's made herbarium specimens knows that that takes a lot of time because that's a really nicely made one for Tripsacum, which is a big, sometimes 6 foot tall grass. So it's a rhizomatous perennial grass. It's got a rhizome down there that's a below ground stem that it can re-sprout from. It's noted for its drought, fire, and flood tolerance in the literature. And you can see here kind of some of that fire resilience where you can actually see this burn part right here of this herbarium sample that they made that I thought was really neat. So after fire, it'll sprout back up and it really doesn't seem too bothered by it. You can find it in cons of prairie too, in burned and unburned sections. It has variable ploides from 2x to 4x, and it's the most temperate relative of maize, like I was saying. So here's what it looks like when you're out kind of in a more natural area. This is the Clinton Lake. This is the dog park. So probably people in this room know a little bit more about this hay field in the back, but I always use this area because I'm out there all the time with my dogs, which are over in the side of this picture. I use this to kind of gauge where the phenology of the plant is. If I'm going out collecting, I can use these as understanding when I should go out. It's a whole lot easier to find Tripsacum if it's in flower because you can see these big, long, wispy flower stalks. So Zea and Tripsacum are sister genera. The split was about 650,000 years ago, although that may be older depending upon what markers you use. So if you use chloroplast or maybe other chloroplast or mitochondrial markers, puts that date a little bit farther back. I think this is a more nuclear phylogeny here. And this is one of the more current phylogenies that I've seen. So pretty closely related, even though they're in two separate genera. And it's still possible to intergress Tripsacum and maize together. So maize being down here at the bottom, Tripsacum being up here. So we're still able to cross those and get fertile offspring. Generally, most people will use some kind of bridging species like Zea perennis or Zea dipla perennis before they intergress anything into maize, just as a convenience because it has a closer background, but you still can cross maize and Tripsacum and get at least something that is fertile afterwards. And so I would say Tripsacum saw like kind of a boom in its research in kind of the 80s and the 90s when it was thought that it might have been the progenitor of maize. So it was thought that maybe we domesticated Tripsacum into Zea maize that we have now. Now we have a better idea of the phylogenetics. We know that's not the case, but it still served as a valuable resource for maize improvement across its time as well. So we've seen, at least in the far one, this kind of plant journal paper, they looked at cold adaptation in maize and found that some of the same genes that were selected upon in artificial selection for maize are the same genes that we see selected upon for Tripsacum in kind of its more northern areas where it's growing, kind of up in northern Nebraska and those type of areas. And then it's been a resource in a lot of maize genetic studies where they've used it as an outgroup and looked at a bunch of, looked at annotating it as well as maize. And then we can see kind of this, this hybrid species that they've also produced. So there's been a lot of research on Tripsacum for its usefulness in maize improvement. And so we've got this big collection. So this is our kind of maize or our Tripsacum dactyloides nursery collection. So we've got a bunch of genotypes that we've collected from all across the United States. And this is in partnership with USDA collaborators who have allowed us to come in and take accessions from their collections. We got a bunch, or Jada got a bunch when she was a early graduate student from Woodward, Oklahoma. And then that collection moved down to Temple, Texas. And we got a bunch of accessions just this last year as well. And we've got these planted out at the KU Native Medicinal Plant Research Garden. So I think a lot of people in the room have some familiarity with it. We're like the people in the back that nobody knows what's going on. So I always try to stop and flag people down and say, this is what we're growing back here. Sometimes, mostly Tripsacum back there, sometimes some other, I think Jay's grown some maize land races as well back there, some of the taller corn looking stuff back there. So we've got accessions from all over the place. ones from the kind of drier plateau of Texas all the way to kind of the Gulf of Mexico in this sea drift sample here. And so what I'm going to talk about today is kind of our work phenotyping this collection. We have all these accessions, we have all this diversity, but we really don't know much about it. We've got some records that we get from the USDA, but they're usually fairly patchy and they don't have any real measurements of what the difference is between two accessions. So I'm going to talk about some work looking at the root anatomy of these accessions, particularly under flood stress. And I'm also going to talk about some of our work looking at stomatal trait diversity as well. So kind of measuring the above ground portion of the plant. So I'm going to start with the roots. And I think in this audience, people understand what roots are, what they're good for. When I go back to my hometown, I tell people I'm working on roots, they kind of understand it. When I tell them I'm working on root anatomy, they don't know where I'm going anymore. So there's a lot more of this mysteriousness below ground, but I try to tell them that if we can improve how a plant produces a root, if it can make it cheaper, if it can make a more efficient root, we can grow a better plant. That's how I try to explain it. So roots primarily used to anchor the plant to the ground. You don't want it to fly away, facilitates water and nutrient acquisition. But there's also a lot of diversity in roots as well. So in basic plant biology, we always teach the differences between monocots and dicots in their roots, fibrous root systems versus a taproot system. So there's a lot of interspecific trait variation and a lot of intraspecific trait variation where we have different types of roots that maybe are serving different ecological roles for the plant as well. And then we can even have more like intra root variation where within a single root, we can have differences in their anatomy as well that might have ecological impacts to how that plant root functions. And so abiotic stressors that I talked about before, drought and flooding, they typically will cause alterations in the anatomy as well. So that's what we're often studying, is looking at kind of these changes. Okay, you can't see my cursor. So changes from different parts of the root. So flooding, which I've talked about, it's becoming more common. It's kind of across different parts of the country as well as different parts of the globe are experiencing flooding at different rates. I think probably what's happening more is we're having just more climate unpredictability, which if you take this week, for instance, where we had a blizzard on Tuesday and it's going to be 70 degrees on Saturday, you can expect that plants are going to have to deal with a very variable climates. So when plants are waterlogged or submerged, their organs are deprived of oxygen. So they kind of have, you can imagine like us, if we're underwater, there's dissolved oxygen in water. We can't take it out of that water because it's harder to pull oxygen out of water than it is out of air. And we're much more efficient at breathing air than water. So when you don't have enough oxygen, plants can't do cellular respiration, they can't generate energy, and their cells are going to quickly die, basically. So you can think of when a root is flooded, right at its root tip, it's growing, it's got a meristem at its root tip, it's growing very quickly. When you cut off its energy supply, the only alternative it has is just to die off. And if you have enough of those roots die off, you're going to have the root system basically start to completely die. So if you've ever waterlogged a house plant, then tried to see what happened. If you ever overwatered a house plant and then tried to coax it back, sometimes it never makes it back to a living plant, I guess is what I would say. So flooding also leads to the accumulation of stress hormones like ethylene. This is one that I study in particular. I'm not going to talk about it too much in this talk, but you can measure this as kind of a level of, or measure transcripts of this to kind of see how much stress the root is under. And these, when they build up, because they're a gaseous hormone, they're normally released from the roots. When you have a bunch of water outside of the roots, it traps it in there. And that can lead to kind of a signaling cascade where it can trigger these kind of rapid alterations of the anatomy of the plant. So why I'm interested in this is that not all plants tolerate flooding equally. We've got two kind of beautiful root sections here, one in rice and one in maize. And we can see we can see a lot of things that are marked, and it looks like something out of a plant biology textbook. But what we see is kind of these big open air voids in the rice root, this wetland adapted species, where maybe you either grow it in a flooded paddy or you use kind of flooding as a weed control system. You see these kind of enlarged voids in the root, versus for maize, we see a lot less of these. So these are root cortical parenchyma. These are these enlarged cellular spaces in the root cortex, which is kind of this area outside of the steel of the root, kind of this area out here. You can see it. I like to say, and this is Tripsacum dactyloides, it looks like a wagon wheel, kind of, is the way I like to describe it, with these little spokes that are the kind of connecting points for the outer epidermis and outer cortex of the plant itself. So the reason why plants form these parenchymatous tissues is that if you form parenchymatous tissues, they're able to better supply oxygen to parts of the root system itself. So we can see this is an older study where they're looking at maize roots. They have the black ones are parenchymatous roots, the white ones are non-parenchymatous roots. And what they find is that there's a difference in the amount of oxygen that's actually in those roots themselves. And you can see it's quite substantial. So like when I'm thinking about flooding, I don't need to buy maize. It's never going to be a wetland species, but if I could buy it 12 hours, 24 hours with just an adaptation to its root anatomy, maybe that would be enough to kind of stave off some of the most detrimental effects of these particular stresses. It facilitates oxygen transport, and then under drought as well, you see parenchyma form as well. And I think that may be due to just lowering the metabolic cost to produce a root. You can produce a bigger root for cheaper if you don't have to produce as many cells. So that may look like something like this, where the plant will take up oxygen and it will transport it through the root. There's also many mechanisms that I'm not going to talk about today of kind of preventing this arrow here, which is radial oxygen loss. As you transport oxygen in the root, some of it will seep out. Plants have a lot of waxes, so if you ever notice like the roots of them having a waxy coating, that may be suberin to try to seal off that root. And there's other ways that it can kind of seal the outer area of the root as well to try to stave off some of that loss. So maize and Tripsacum differ in this key phenotype. So we can see examples here. These were taken by Nicole Ginion, a postdoc in the lab. They use this kind of laser ablation tomography technique. It produces these like very cool images. They're really small and maybe a bit hard to see, but I'm sure the Zoom can see them well. And we can see the big difference between Tripsacum and maize. Tripsacum, even though it's a close relative to maize, has copious amounts of large parenchyma, whereas maize has a smaller proportion. So Tripsacum generally constitutively produces root parenchyma, whereas maize, most of its root parenchyma is produced or induced under stress. And so I like to think about like the tortoise and the hare. Like Tripsacum is already producing these air voids under well-watered normal conditions. Maize has to kind of play catch up always because it goes under stress. It has that stress triggering of the buildup of ethylene, produces these parenchyma, but maybe it has a lag period where it's not able to supply any oxygen to the root tips and then maybe die off. And we can see here just the representation of the data here over a larger number of plants. You can see that Tripsacum has maybe about double the amount of parenchyma under normal conditions than we see for maize oftentimes. So root cortical parenchyma, it's a heritable trait, means that we can breed for it. They've found inbred lines or rills of maize that have higher or lower amounts of root parenchyma that they produce. It's also ecologically relevant. Here they're looking at it under well-watered conditions in the black and water-stressed conditions, basically drought in the white bars. And you can see that they have lines that produce more root parenchyma. In that study, they also found that they were able to produce bigger biomass under these same conditions as well. So it's heritable and it's very ecologically relevant here. So we can see a better example with some nice pictures where we can see they took a wild type line of maize, this one on your left-hand side. They took a tropical wild relative of maize, this is the Nicaraguensis. They integressed a QTL for constitutive parenchyma production into maize. And then you can see the ones that I highlighted are the maize wild type line and the maize integration line under stagnant conditions for 14 days. Stagnant being low oxygen, which here they're growing in hydroponic systems, but what you can see is that the plant is better able to survive those kind of low oxygen environments with the amount of root cortical parenchyma it makes in that integration line. So that was a good proof of concept that this is an interesting trait and worthwhile to kind of profile in our collection. In Tripsacum, I figured it was going to have a ton of parenchyma because there's a ton of anecdotal and more colloquial knowledge that it's very flood tolerant. So in the flood of '93, they've got some genotypes that they said survived that entire time in Ellsbury, Missouri, at a USDA facility, 23 days of inundation of water. And then I've also seen for more millennials, even on Instagram and stuff like that, people will call it Tripsacum as a nice prairie plant species, and they always usually note that it has both flooding and drought tolerance. So that's something that in our lab we've been trying to study. So what do we know about variation in root parenchyma in Tripsacum? Well, very little. A lot of the research in Tripsacum is from the '90s. It's from an earlier time where it was thought to be the progenitor of maize. So a lot of research went into it at the USDA. A little bit less now. The USDA is more concerned with Tripsacum for a forage crop than as something as a primary resource to improve maize germplasm. So what they found in this study was that Tripsacum, this is just a qualitative study, does it produce parenchyma? And that's what they found here is that for our Tripsacum dactyloides, our accessions they took accessions from Texas, Kansas, Oklahoma, they all produce parenchyma. And then they looked at some other Tripsacum species as well as varieties as well, and looked at if they form or are able to form parenchyma. So we've got a little bit of work to build off of, but nothing quantitative that we were kind of looking for. So that's where we went to our kind of diversity panel that we had. So we have all these collections from across the country. And we have this perennial species that's easy to replicate. We can easily take one genotype and make multiple more genotypes. So that's what we did out in the field site. We set it up out there. We collected clonal ramets of the Tripsacum by harvesting them, mostly with a shovel and pickaxe, breaking them off. We took a bunch of measurements of those ramets, and then we grew them out in the field, or let them root out in the field in those small containers for a month before we brought them into that's the Hayworth greenhouse right there, where I put them under waterlogging stress for 72 hours. And then we took a whole bunch of phenotypic data, and that's what I'm going to talk about today, and kind of how we did that. So kind of one of the problems that we had with this collection is that we collected a ton of roots that we wanted to section. We had over 200 accessions that we have in our collection, and sectioning via light microscopy is quite tedious and slow. You have to cut a very thin section. You need to stain it. You need to then mount it on a slide. Usually we're wet mounting it with water, so it's a little bit easier. And we're working with embedded tissues, so that's easier too. But it was still quite slow. And I was thinking that we're going to have to be a little bit faster if we want to process this amount of data. And I'll stop kind of here to thank the sectioning experts. So they still made, I think we've made probably 600, maybe over 1,500 sections now that we've got root phenotypic data from. So they're now, I trained them, but now they're more experts at sectioning than even I am, and much better at it. So one of the ways we looked at speeding up this root imaging was to use kind of a different technique. So instead of making a section of the plant, what we do is we use a microscope that's going to shine light at the root, and we're going to catch the light coming back. So instead of shining light through some sort of tissue, we're actually using more of a kind of like pseudo-fluorescence imaging. So roots are naturally autofluorescent at 375 nanometers. So that's what we looked for. And this was from a tip from some researchers at the Danforth Plant Science Center. They kind of were showing that these were useful, and we've kind of taken this idea and tried to run with it a little bit. So we're imaging from the cut into the root. So instead of making a section and shining light up through it, we're taking a root, we can lay it out longwise. We can make just a parallel cut so it's got a flat surface, and we can shine the light at it and get it bounced back. So then we can kind of make our sections just along the length of the root faster without having to make them very thin, which tends to be difficult to translate to somebody else to try. So it also is nice because it's very cheap. but it also allows us to image these roots that typically are very hard to actually image, because you can imagine trying to make a section of this root down here, which for the zoom, this one, it's undergoing root cortical senescence, where the cortex of the root is basically going to slough off. So trying to make a section of this is very difficult, because usually what happens is you get your steel and your cortex of the root, they just float apart in your water when you're making the section of them. So it can allow us to kind of section materials that are oftentimes, well, they're all unembedded, and they're all usually sometimes a bit harder to actually phenotype. They're a bit harder to get a clean section of. And you can see here, we haven't stained the root at all. We've got just an unstained root, and it's just showing autofluorescence. And you can even see some of the tissues of the root that are generally lignin, have a lot of lignin in them, actually show up a little bit brighter. So I think there's more phenotyping that we could do with these sections as well, but I'm not going to talk about that too much. And this is our setup: a little laptop, a couple of stands, a couple of 3D printed parts that we use to hold the roots, and just your basic lab equipment. So about maybe $2,000 once you talk about a computer and the stages and stuff to hold the microscope steady. And we can take a bunch of sections really quickly, and I loaded up a bunch of the sections that we're here to take, and you can kind of just see all of them here. Varying degrees of fidelity too. So what kinds of data can we extract from these images? Well, we can extract basically anything that we did on the light microscope. We can get areas of different parts of the root. We can look at the arenchyma, which is what we're concerned about, metaxylem. We can look at the kind of thickness of the epidermis, and we can also count the number of cortical cell files that can either produce bigger or smaller cells in the cortex, which is what we're interested in. We can take those measurements and derive a bunch of other indices as well that we're interested in looking at. If anybody wants to know about the image analysis part, I don't have anything on it really in here, but I can explain that. We use ImageJ and open source software to do all that image analysis. So it's low cost and low effort, but it wouldn't be for anything if it was very inaccurate. What we find is that it's very similar to the light microscopy. So we just took a set of samples, imaged it on our UV microscope, and then imaged it on our light microscope, and made sure that they were kind of corresponding to one another. And we can see these kind of lines showing us the one-to-one, that for the most part, they're fairly accurate depending on, regardless of what method we're using. Some of them are a little bit more skewed because they're a little bit easier to count the number of cortical cell files if you have a very thin, transparent section. And happily for me, we're moving at 20 to 25 samples an hour versus 7 for the light sectioning. So we're moving much faster, which means that we can include many more plant species, and we can take sections across the length of a root as well, because we can actually scale up to thousands of samples when we're doing 20 at a time. But I think that we can get that a little bit faster with dealing with some of the issues with naming of files and stuff like that. So some of the not fun part of taking the image is naming the files. So we're trying to implement some barcodes and stuff in there to make it easier. So with that, we're looking at the arenchyma again. So I've got just these plots here where we've taken sections of the root, and we're looking at the base of the root in the top, and then 3 sections from closer to the root tip. And here I've got them broken into just deciles. I like these graphs because I can kind of look across and see how it's changing or how it's shifting across the percentages of the amount of the cortex that is arenchyma. So that's showing all of the samples there, or that's representing all the samples there and breaking them up. And so I can start to look at, I can take an accession like this one, McCain 334-11. This one's from Mississippi. And we can look at where it falls out in these kind of ranges. We can compare that to another one. This one's from Caldwell, Texas, a little bit east of Austin. And we can look at where it falls out on here. And what we can see is kind of accessions that have contrasting amounts of arenchyma that they're producing. And so then I can look at these, and we can see these kind of stunning differences that we see in the amount of arenchyma that we have for different accessions. We can even see cases where if we were to just take one section and with a slower method, we may not get an accurate picture, because at that sixth section, we had a little bit more parenchyma for the Caldwell sample than we actually had for the McCain sample. So I'm trying to use these as kind of more of a composite than just, traditional anatomy might take one or two sections of the root and label that for a genotype. I'm trying to see if we can make more sections along the length of the root and maybe do some modeling to say, how that root, interpolate between those points to see how that root actually is constructed for a given genotype. And so with that, what I plan to do with that, we have a bunch more data, I should say, a bunch of root trait data for the root architecture and root structure as well that I can load into a PCA, get measurements out as well. But the plan for these is we want to take a subset of these accessions and replicate them in a microbiome exclusionary experiment. I've seen through working with corn and inoculating it or keeping it under sterile conditions, I've seen differential performance under flooding stress. So I'm thinking microbes may contribute to a plant's response, particularly because some microbes can actually work to degrade precursors of ethylene. So they can maybe help dampen a plant's flood stress response by using up ethylene, or there are certain microbes that can make kind of toxic compounds for the plants when they go under these kind of stagnant or hypoxic or waterlogged conditions. So that's the plan for that data, is where we're going with that. We needed kind of that baseline to say, do we actually have interesting phenotypes out in the field that we can further study in kind of a more replicated experiment where we can set up these hypotheses that ones with more parenchyma should do better under flooding, ones with less should do correspondingly worse. So put in the slide to switch over to stomata. So now we're looking at the above ground part of the plant. So that was the root part. We're looking at these same accessions, but now we're going to look at the stomata of the plants. So stomata are these pores on the leaf of the plant. They can also be on the stem too for some plant species. And they facilitate gas exchange. They're really small, and we can see that it's going to take in CO2 and give away oxygen. And every time a plant opens or closes these stomata, it's also losing water. So it's costly to open these. And so it's one of the things that plants will close up when they're under a drought stress. So to look at these stomata, because they're very small, oftentimes what we do is we just make an impression of the leaf. So we use a resin or a wax, or you can use different types of putties. In our case, we're using 99-cent nail polish that I bought from Walmart. We tested a few different varieties, and some have some different properties. We want the speed drying version. What we do is we paint it onto a leaf that you can kind of see here. We peel it back up. We usually use tape, and then we tape it onto a slide, and we can image that on a microscope. And I've got a few pictures of undergrads in the lab collecting these samples out in the field, where we just take multiple leaves, we tape them to our table and do them really quickly. And then I've got down here, Kara was a former undergrad in the lab who went on to the USDA, and she's training a couple of other undergrads how to make these sections on a maize plant that we brought up from the greenhouse. So this is what our impressions can look like. You can see they have a lot of different structures. Here's most of them labeled. There's still some that I'm unsure as to what I'm looking at, because I'm looking at an impression of the leaf. So that top one, some kind of hair or pit. I'm not exactly sure what that is. We've got for grasses, a lot of them will have these bulliform cells, these cells that allow the plant leaf to curl, kind of like a taco, which is good under drought settings, to be able to curl your leaf, maybe conserve water. And then we've got our short cells, where we've got these silica, these phytoliths. We've got long cells. They have botanical terms for their shapes as well. And then we've got kind of the star of the show, the stomata that we're looking for. So what we have is a bunch of images, and we need some way to classify them. So the way that we do that is through object detection, or kind of these machine learning or kind of computer vision models. So the model that we used for identifying our stomata, we didn't want to have somebody hand annotating every stomata, because it gets really time consuming. So we use this kind of YOLO model. It stands for you only look once. Basically, a lot of other computer vision models look at an image iteratively. And if you have 1,000 images, maybe that's not so bad. If you have 1,000,000 images, it starts to get really time consuming to look at something over and over again. So these ones are kind of more state of the art. A lot of companies use them like Facebook, Ultralytics, and Microsoft, and they release a bunch of open source code that we can use that saves us a bunch of time because we're not starting from a completely untrained model. So that's what the model looks like. I can't explain the math behind it. It's a bunch of convolutional neural networks, a bunch of machine learning. But I can apply it to what we're looking at. And a lot of the aspects, I should say, of what Facebook and all these places are looking at, they're looking at it for video detection. You can only look at it once because it's live. A lot of times they're trying to track things in live security cameras and stuff like that. Ring doorbell cameras is what you can think of. But we're going to train this type of model to detect stomata and leaf imprints. So the first thing that we have to do is we have to give it a data set where we have stomata that we've hand annotated. We use this program that's an online resource called RoboFlow to annotate these stomata here. Then we can export these kind of as a data structure that the model can take into and then learn from. And so we manually annotated 70 images. That was 3% of what we generated. It took about an hour to train the model. It's training all these different parameters, what it's looking for, how much confidence it has in particular predictions. And then we compared it to hand counts that Kara made on a subset of images. So we sent it off, said, annotate these images. I compared it to what Kara got, and we got about 90% accuracy, which is probably, as we could probably get that up a little bit if we gave it a bigger data set to train with, if we put in images that had things that look like stomata, but we've like things that have some of the things where it struggles with prediction, these parts of the leaf that maybe are a bit harder to actually image. I'll show it in another one. So collected 138 plants, 3 leaves per, looked at the top and bottom surface, the adaxial and the bottom surface of the leaf. We took 3 fields of view. Then you can see our replicates here, where we've got the top and bottom part of the leaf, we just put on the same slide for convenience. So we ended up with 2,400 images, or a little over 2,400. So I'm going to ask the audience, how many stomata do you think we had overall?
What's the area of a single image?
0.031 millimeters squared. I know that because it's on another slide. Like, I'm pretty sure. 50,000 more. You didn't go over, so I think that's Price is Right. Still good. Okay, I'm
About 250,000.
No, less. 84, about 84,000. 86,000 were annotated. We cut some of those back because the model had low confidence in them, so they probably were getting something wrong. And I do notice things that it gets wrong, but your images come back looking like this. So they come back with kind of these predictions of the stomata on them. And you can see it even tags them with a little confidence rating. 1 would be, I'm totally sure this is stomata. 0, we're not even seeing that because I throw away everything under about 90% because it's usually picking up on stuff like hairs, little occlusions in the leaf, or damage in the leaf. So we get from these measurements, we not only get the density, 0.3311 millimeters squared. Sorry, I got a decimal wrong. I had seen this before. I had to measure it a few times because you want to make sure what size you're measuring. So it will also clip out and give me 84,000 of these little stomata boxes too. So I can actually measure the size of them. It gives me every single image. So I think if you were to iteratively train this, you would overfit it to work well on Tripsacum, but you could probably get it to work more accurately on Tripsacum by feeding back that 84,000 into the model. So we see differences in the leaf surface, and that's obvious. We expected this, and this is kind of good that we do see these differences where the bottom surface of the leaves, at least for Tripsacum and many plant species, have more stomata than the top parts of the leaf, the adaxial, the green ones. And we don't see any real difference in the size of the stomata across these different surfaces of the leaf. So that's good that we're not seeing something kind of crazy going on. Most of what I'm going to talk about today is the abaxial part of the leaf, and a lot of the other plots I'll show, but the pattern is pretty much the same for the top or bottom part of the leaf when it comes to the kind of genotypic effect. So we took some data that our collaborators down at the USDA in Temple, Texas had collected previously on Tripsacum. They were characterizing some of these ecotypes, and they looked at leaf width, kind of putting it into these qualitative categories: broad, intermediate, or narrow. We have one that's unknown as well. So here's Jim Canary, here's Amber Williams, both formerly at the USDA in Temple, Texas. So we took this data and wanted to see, we had this data on hand. I wanted to see, does this correspond to anything that we're seeing with our stomatal measurements? And I was kind of blown away. And we actually do see like a kind of very stepwise pattern where these narrower leaved accessions have higher densities of stomata in smaller stomata overall, whereas our broader leaf ones have lower density and larger stomata. Let's try not to get tripped up on that. So I have a bunch of plots that'll have the density and it'll have the area as well. But we see this pattern where these broader leaves have these bigger stomata, but have less of them. So picking out two accessions to illustrate this point, we have this accession from Bartlesville, Oklahoma, and one from Jackson, Texas. And you can kind of see that relative difference in the stomatal size. You can also see, I'll point it out with the mouse, stomata that we missed. So our model's not perfect. I mean, we're still missing some. So like this would probably go up because I can see one there, there, and there. Probably because we have this little either bubble in the resin or the nail polish impression that we made. But I thought that was really cool. But I think there's a big grain of salt that you have to consider the ploidy when you're looking at stomatal measurements, because if you have, this is my grain of salt, that we don't know this pattern all the way, and what we saw there is cool, but it doesn't tell the full story. So ploidy is correlated with stomatal size because you increase the minimum size of a cell and you increase the size of a tissue and an organ as well. Here's an example in an African periwinkle. They mutated it and made a triploid or a tetraploid that they looked at, and you can see all of the stomatal measurements as well as are larger and the stomatal density goes down. And where we're at now with what we have ploidy information or ploidy information for our accessions, you can see that our tetraploidic accessions, which should have larger stomata, are more the broadleaf ones, and our diploids are more these narrowleaf ones. So what I showed you before, you got to take it with a grain of salt because we need to understand and control for ploidy in the model. And at the moment, right now, I don't have the ploidy for a significant number of the accessions that we're looking at. We can look at the data that we do have for ploidy, and we can see patterns, kind of that stepwise pattern. I'm thinking also that we can kind of annotate, maybe we can give a prediction of the ploidy just from the size of the stomata that we see on some of these impressions. So you can see here, we have this pink unknown category. We have our diploids here. We have a lot that kind of fall in the bounds of where this diploid is. So maybe if we measure enough stomata, we can make a pretty accurate guess as to, do we have a diploid or I think we could probably predict a diploid and a tetraploid. The triploid falling in the middle is a lot harder to get. So one of the ways we're going to ground truth this is we've got some population genetics analysis underway where we can look at allele frequencies to determine what ploidy it is. And then kind of our gold standard, which is using flow cytometry. And this is Braxton has been validating a protocol for extracting nuclei from Tripsacum to kind of get at a kind of method to determine the ploidy for all those pink ones, all those unknowns. We really need those before we can say much more. We also connected this. We just looked at how this was impacted by climate. So I pulled a couple of bioclimate variables, bioclimate 1, which is annual temperature, and bioclimate 12, which is annual precipitation. I put those on the chart. I looked at how stomata size changed across these different bioclimate variables, and we see that as you increase the temperature, kind of increase the aridity, you see an increase in stomatal size, which has been found in the literature too when they're looking across a lot of species. As you go into more drier, hotter environments, you have larger stomata, maybe because you can better control your water loss through bigger stomata. So we're seeing some ecological relevance to this as well. So I think that's pretty much where I'm going to stop. I'm going to plug Jada's defense, which is coming up. I think that some of the work that we're doing with these stomatal traits may help explain some of the differences in performance that she's seen under some of her rainout shelters, and then some of the microbiome work that she's done, seen interesting patterns with the stomatal density and the stomatal, I think mostly density, not size. And then I'm going to go forward with this work. I've got now these accessions that are kind of on my hit list to test in a more replicated experiment where I can see, does increasing the amount of root parenchyma confer greater flood tolerance? It definitely should, from everything I've read from the literature, but we can really test that. And what I really want to get at is how these differences, especially in the cortex of the root, impact the microbiome and impact the ability of the plant to form associations with particular microorganisms. So there's been a little bit of research in the past that's looked at parenchyma and how it impacts pathogenic and beneficial fungi as well, but not much to follow up from that. So I think there's still a wide open field of considering. I didn't just look at a root microbiome. I looked at a root microbiome with this type of porosity in this particular portion of the root. Maybe that would help explain the patterns that we're seeing. And with that, I think a little over, but I'll thank everyone at Kansas, USDA. Tom gave me access to his root silhouette box. I didn't take a bunch of, I didn't show a bunch of that data, but we generated a bunch of it. And then Sheena, Nathaniel, and Bruce out at the field station have helped with mowing and tilling and kind of helping set up this big this big nursery plot that we continually add to. And with that, I will thank all of you guys. Okay.
Can you see the question?
Yes, I'll read them too because they're probably pretty small. Where does teosinte fit in the phylogenetic tree in the early part of the presentation? Maybe I should just go to it, but teosinte would fall somewhere closer to Zea. I'm not exactly sure. I think that tree probably would have included it too, but I'd have to go back and look.
It was there. It's a subspecies. It's called Zea mays parvicularis.
Yes. Yeah. Okay. Good point. Yeah. Jada has the name for it. I was gonna say I've mostly looked at Tripsacum and maize, so it's like that's where my head's been at for the longest amount of time. Okay, so this one. When I see plants in the field that are so huge, curious if stomata or root characteristics vary from the edge to the center of the plant. I realize this isn't an issue with your samples, which are similar size, but perhaps increasing the growth of plants in natural ecosystems. I think Helen hits on an interesting point, especially for stomata. We took it at one time point. I want to take it at a couple of different time points, especially now that we can quantify it faster. We were able to collect all 138 times 3 leaf impressions in a single afternoon. You can really, once you have a workflow for doing it, you can collect those slides relatively easily. I think we can begin to assess, yeah, if there's some kind of seasonal pattern. I think we tried to standardize on a particular leaf, a particular area of the leaf in a particular type of leaf in Tripsacum. It'll have these big flower stalks and it will also have leaves that come off the flower stalks. We're looking for leaves that come off the tiller of the plant, so kind of towards the base. But I think that there's going to be variation within a plant, and I think that it's possible that there's some variation within the season of a plant. But I think stomata traits are probably more genetic in origin. Like this genotype produces this type of stomata, but happy to kind of look at that more. With the root characteristics, it's a little bit hard to collect, so we often only get one time point, the ending time point in most experiments. And it's destructive, so we kill the root. So we're always having to have more samples because we're sampling from a different root each time if we're working on a single plant. Just a comment. Rule of thumb is 3 days with roots underwater. Corn doesn't come back, but cooler temperatures help prolong this. Yeah, so he's hitting on the point that roots, when they're flooded, actually some of the most detrimental effects of flooding are after the flood is receded. The plant struggles to regain that water balance, and it really struggles to take up nitrogen. So a lot of what I've seen is that when you have flooding, what you do afterwards is you dump a bunch of ammonia fertilizer on it afterwards to try to bounce it back. But I mean, if we could just do better in the flood, maybe we don't need all that. Any questions from the science? Yeah.
So you talked about the influence of ploidy on stomata.
Yeah.
So have you looked at that with respect to root anatomy also? So
Yeah.
that can affect, cell size?
I think it could, it could affect cell size just because that minimum cell size is getting bigger. And one of the things that we don't have a good way to phenotype is cell size in those root sections. Even though we can see it, like I can show images where it's super easy to see. You know, I mean, like probably got a bigger one somewhere. Either way, bring it to the front. We can see these differences in cell size. It is really hard to get a computer to annotate that. I've struggled. I don't have a good, I've tried to make the same model that I made for the stomata to analyze these. It always is getting things wrong. I think because everything's kind of like a circle. So if you're working off shared characteristics, the computer doesn't know anything about the boundaries of a root where things should be. So it's just saying, this is a similar size circle. So that's why we count the cortical cell file, but I think it's a very poor estimate of cell size. We need to just probably hand measure some of them.
And just out of curiosity, what percentage of the accessions do you know the flooding level?
To a degree where we have evidence from flow cytometry, it is, you know, we still have a lot of unknowns. Unfortunately, yeah, that it tends to be a bottleneck. That's why we're working with there's a flow cytometry core at KU now in Hayworth. So we figured that's kind of the gold standard to, unless we want to do like chromosome counts, which we just don't have any, I have no skill in that. I've heard about them, but I haven't done them. So if anybody has experience with that, I'd also try that as well. Because we have the accessions, we can easily go out and get material at all times to kind of do these types of techniques, but we're still missing a bunch. Ben, you have a good question?
Yeah, man, those are amazing images. That makes you blows my mind of all the things you could apply. I guess I'm thinking about functional trade-offs, and maybe you talked about there with dumping nitrogen on.
Yeah.
So when you have more parenchyma, there's got to be a cost to that.
There's a cost at taking up water. Root's less efficient at taking up water because if you think of that outer cortex of the root, most of that movement of water and nutrients is what, like apoplastic. So like it needs the connections, right, to like move it to the center. So there's some trade-off where it gets so porous that the root doesn't do anything, that cortex doesn't do anything. And up in the base of the plant where maybe it has more porosity, that root really isn't taking up water anyway. So maybe your root tip still has to be pretty solid where it's actively taking up water, but that's, I think, kind of the trade-off. If you have way too much parenchyma, you're going to suffer in your ability to take up water.
So if that's one of the important trade-offs, I mean, I honestly thought you were going to have a, excuse me, a third part that tried to relate stomatal density and your parenchyma, right? Because we tend to think of plants as big straws, right? So
we've got it for the genotype level. I think we could. It's always like, how do we connect these two? And then I always try to look at like, is there literature to say that I should try to connect these sometimes? Maybe
just how plant, how evapotranspiration works. I mean, I think is a baseline enough, right? I don't know what you would predict or whatever, but it seems. I mean, of course, I love microbes, and I want you to manipulate microbes and see what happens. But I think even without that, just from the data you have, you could see for a given accession, you have like repeatable relationships between stomatal size or density and parenchyma density or whatever the metrics you had.
Yeah. I did put all of them into a correlation matrix, and a lot of the stomatal traits were not correlated with anything but stomatal traits. Okay. I think that's telling me that. So that's where I was like, okay, like, how do you get it? When you have a bunch of traits and you don't know where to go, it's like, I'll look at a correlation matrix to say, like, is there interesting patterns that are outside of all the root traits or all of this traits, like connections? Difficult. I wish there was more.
I mean, if they're genetically independent, that's interesting too. Yeah. Like for a plant, like how a plant moves air and or water, right?
Yeah, a lot of phenotyping still to do.
You might get a different answer if we use the genotype means to do that correlation matrix. If
there's
a lot of plasticity, it could be obscuring something interesting.
It seems like in the stomata, it's more consistent across the genotype, but then the parenchyma is less consistent. I mean, that's why I want to make it faster so we can have more replicates, because it seems there's a lot of choices that you make when you're selecting which route to look at, how many sections as well. There's another question somewhere. Oh, in the back.
So could you speak to what you found in the literature about the microbiome microbiome. So what are you seeing in literature?
Yeah. Here's the junk I cut off. Not junk, these are great studies. I just don't have time. In this study here, delete that. They found as they increased the amount of porosity in the root, they found that AMF colonization went down and pathogenic fungal colonization went up. It was kind of in opposite directions within corn, and using a kind of different technique where they could actually they think they're imaging kind of these arbuscules there, but I can send you this paper. I think this paper is really nice, and I'm hopeful that that method where we're shining light at the root. I need roots that have AMF, that an expert can tell me like, yeah, these are definitely AMF, because I can, maybe we can image it, but I'm not an expert in fungi. So I think that with the right data set, maybe we could use this same fluorescence technology to look at colonization of fungi. Bacteria, we're not going to see, they're too small.
So we don't know whether or not AMF might confer some resistance to inundation.
Oh, yeah, I'm not exactly sure. There's a lot more literature on AMF for drought stress. And there, yeah, definitely, it has usually a very positive influence on plant water relations under a water limitation, just from the, I mean, you can think of the size of a hyphal network is going to be able to take up more water, but a little bit less. And a lot of flooding research is based on hydroponics. So I'm also like, how much of this can I relate to the field? And I have to grow them in pots, and they're still big plants. So there's still some questions, I guess, as to like translating some of what I've seen in the literature across. Another question?
Well, I had some speculation. Been two similar questions probably, but at least, but the idea that there's how many stomata area or spacing is good for hot, dry, sunny days versus,
Yeah.
I mean, and you only have like one little tiny point suggesting something,
Yeah.
you know, like, or versus flooding, or is that bad? That
I'd love to
is
measure
the tetrapoid going to give you too big a spacing between the stoma, or I don't know. How do you know?
Yeah, so like I guess a phenotypic trade-off between density and area, and you definitely see they're always kind of going in opposite, or they're negatively correlated with one another. Yeah, I think that this gets to at least some of the literature that I've seen on maize, where they have what they call like ideotypes, like or phenes, like collections of traits that they think would do better under a particular region. And they think, okay, we stack up these traits, and then we put this into this particular region, it should do better. But yeah, how much we know about the actual trade-offs is always kind of lags behind. We have to like find the accessions, then you got to test them. And I think you hit on a point that we tested one portion of a leaf. If I test a leaf that's on a flower stalk or a different part of the plant, I mean, Tripsacum is, we try to standardize, but yeah, I think that we could see some different patterns depending upon what we looked at. Leaves on the flower stalk are there as well. We didn't test those. We tried to avoid all of those. And we tried to select similar sized plants. So that's why we ended up with a little bit, we didn't phenotype some of our smaller plants. We wanted to try to keep it, you know, ones that are relatively at their, I don't want to say maximum size because they grow really big, but they've been established for several years now at our field site. So I don't have a good answer for the question. I don't know. That's kind of what I want to study is, can we now manipulate them under a drought stress? If we understand the ploidy, and I put my most contrasting large stomata and small stomata plants into a drought stress, know that I have the same ploidies, do they actually perform better, or do they both deal with it in different ways? I think it allows us to kind of generate a hypothesis that we could feasibly test, which is what I was trying to do with building up this data, to try to get something that we could do something with it. I think that's all for the chat. Any other ones? I'll be around too. I mean, we have methods for doing some of this. I think that's useful if you guys have data sets you want to try it on. It's not usually, especially if you're doing sectioning, it's not super difficult.
All right, let's thank Joel.